Okay, quick review of what we did yesterday. Why do cells divide? From page 184, we said there were 3 answers to this. They have 3 reasons: reproduction, growth, and repair. And so we're going to continue today on page 185. On page 185, our question is going to be, what happens to the genetic material during cell division? It says genetic material in the cells is called DNA, or deoxyribonucleic acid. It contains the information that determines the traits that a living thing inherits. And in eukaryotes, DNA is found in the nucleus. But what about in prokaryotes? In prokaryotes, the DNA is free-floating. So add that to your textbook pages. When we have a eukaryotic cell, the DNA actually wraps around, here's our double helix DNA, it actually wraps around proteins to form what's called a chromatid. And the chromatids are like the legs on the chromosomes, and they're attached in the middle with a centromere. Before a cell can divide, it has to have a copy made of the DNA so that both of the new cells have an exact copy. So we're going to write our question. Our question, what happens to genetic material during cell division? That comes from page 185. We're going to add some information about DNA. DNA actually stands for deoxyribonucleic acid, so DNA. That DNA contains information that determines our inherited traits. The DNA wraps around proteins. We call that chromatin. And chromosomes are described as thread-like structures. They're specific lengths of chromatin. Specific lengths of chromatin. And then we're kind of use a little shorthand here. Before the cells divide, symbol for division, DNA is copied or duplicated. Then the chromosomes condense, and they condense into something that looks like this. We have 2 chromatids. Here's 2 chromatids, and in the middle is a centromere. Moving on to textbook page 186. What are the stages of the cell cycle? In red, you can see that we have interphase, mitosis, and cytokinesis. And so we write our question, what are the stages of the cell cycle? And then we will write our answer. After reading about interphase on page 186, it tells us that this is the phase where the cell is not actually dividing. The main thing that has to happen during interphase before we can have cell division is that the DNA has to be duplicated before cell division. Now, when we talk about cells, most of the time they are in interphase. This is the majority of the time they are in interphase. Very important, put a star by it. Changes that prepare a cell for division. Before a cell can divide, the DNA must be duplicated so that each new cell gets an exact copy of that genetic material. On page 187, mitosis, it says in eukaryotic cells, mitosis is the part of the cell cycle in which the nucleus divides. Prokaryotes do not undergo mitosis because they don't have a nucleus. Mitosis results in 2 nuclei that are identical to the original, so that the 2 new cells formed after cell division have the same genetic material. The chromosomes condense during mitosis, and in many eukaryotic cells, condensed chromosomes are visible under a light microscope. Here we see the chromosomes that are being pulled apart for the cell division to occur. So in our notes, we're going to write mitosis. The nucleus divides into 2 identical nuclei. Nuclei is the plural form of nucleus. The chromosomes condense. Cytokinesis is the 3rd stage, and this is where the parent cell cytoplasm is actually divided. the cell membrane starts to pinch inward toward our two new nuclei, eventually pinching all the way to form complete cells. If it is a plant cell, there is a cell plate that forms, which will eventually become the new cell wall. So on your textbook page, label this as the cell plate. When we look at our notes, we have cytokinesis is the division of the parent cell cytoplasm. In plant cells, a cell plate forms, and it ends with two identical cells. On to page 188 and 189, it says, what are the phases of mitosis? And then in red, we have those phases. Prophase, which it says the chromosomes of the nucleus of the cell condense and the nucleus starts to break down. Metaphase is when the chromosomes all line up in the middle of the cell and the centromeres of the chromosomes are the same distance from each side of the cell. Anaphase is when the chromatids separate and they're pulled to the opposite sides of each cell. And then we have telophase, which is the last phase of mitosis, and the nuclear membrane forms around each of those new nuclei that are two identical nucleuses, and the chromosomes become less condensed, and the final stage of cytokinesis begins during telophase. So I can tell that I'm going to run out of room here, and I'm going to just change my summary to a little bit later. I'm going to write my question as what are the phases of mitosis, and I'm going to put a 4 out here because there are 4 phases of mitosis so that I can write my answers. And my answers are prophase, metaphase, anaphase, and telophase. I'm going to write more about what happens in each of these phases. So the main thing in prophase is that the chromosomes condense and that the nucleus starts to break down. In metaphase, the chromosomes, which are looking like these little X's now because they've condensed, the chromosomes line up in the center of the cell. In anaphase, we actually split the chromosome into 2 pieces called chromatids, and those 2 pieces start to separate and go to separate sides. This is identical to this, and so we have the genetic material where it will eventually be part of 2 different nucleuses. And then finally in telophase, we have our new nuclear membranes beginning to form around the genetic material. Our chromosomes inside those 2 nucleuses begin to start to unravel or become less condensed, and the cytoplasm actually begins to separate. So I want you to think of a summary that you could write, probably 2 sentences, and it needs to answer the how do cells divide question.